Today we will discuss about autophagy and starvation. So we must have heard about that intermittent fasting might trigger this autophagy. So what is this autophagy means and how it will trigger during starvations? We will cover that thing in brief. So we will start with the autophagy. So in the autophagy, when the organelle that is uh, present within the cells, for example, mitochondria is the organelles. So the organ of the cells are called organelles. So when these organelles get uh, infected or in infected or death, then those organelles need to be recycled, and the recycling of those organelles is being performed by a process that is called autophagy. So how it is done? There is that organelles it's been surrounded by autophagosome in the cytosols in the or cytoplasm, and after that those once it is surrounded by the autophagosomes, then those that organelles or infected organelles will be transferred to the lysosomes, and thereafter the fusing of these lysosomes to this autophagosome occurs, and that is shown here. And once this fusing is occurs, the enzymes that are present in the lysosomes will disintegrate those organelles and recycles the amino acids that can be used again in the cellular process. And it's the final step, that is utilization of the degraded product. It, that amino acids will be used for the protein formations or on the other cellular process where amino acids are required. This is a basic overview of how the autophagy works. And now let us understand how during starvations this autophagy get triggers. During starvations, the two uh, things occurs. First one is that this AMP K pathways get triggered, or and then second one is that this mTOR C1 get detached from the cell membrane. And once it is detached from the cell membrane, what it will does is it will inhibit this Bacillin complex formations. At the same time, this UCL complex formations is also get inhibited. However, the AMPK pathways acts as a positive signals towards autophagy. So what it will does is AMPK pathways will form ULK complex, and once this out of this once the competitions between AMPK and mTOR C1's pathway, in the competition of AMPK and mTOR C1 pathway, when the AMPK gets uh, AMPK pathways uh, gets triggered more, then this UCL complex net UCL complex formation occurs, and once this UCL complex formations occurs, then it will trigger the autophagy initiation, that is ATG complex formation occurs, and this similar process that we have just discussed will starts to form. That is, once the ATG complex formation occurs, it will form a phagosome formations, or we can say that it will try to cover the organelles in the cytoplasm that are dead or that are non-functionals. And once those autophagosomes will engulf all these organelles, non-functional organelles, then it will dock to these lysosomes, where the enzyme in the lysosomes helps in the degradations of these organelles, and the amino acids can be recycled. And these things uh, occurs, that is AMPK pathways triggering, and this mTOR is detaching from the cell membrane occurs during starvations, and because during starvations there is a deficiency of the nutrients, and this will finally leads to this death of this organelles or the inefficient functioning of the organelles present within the cells. So more the starvation time, the more will or the autophagy will occurs. However, the too much nutrient deficiency might cause too much autophagy, and that will leads to that will leads to the decrease in the amount that will lead after some time. What will happens is that all the even after recycling of the amino acids, the amount of amino acids are not sufficient for the normal functioning of the cells. That's why the starvation needs to be occurred in between a certain range, and those range varies depending on the person to persons. If starvation is too long, then it will cause a problem such as the prolonged autophagy, and that is not beneficial to the human beings. So I hope you got an idea about this autophagy and starvations and how it will act at the cellular level. If you need to understand more about these topics in detail, you can go through the references that has been provided in the description, and also you can see the references in the uh, at the below of each figure. So that's all for this video. Thank you for your time.